investigator at the NIH, and she's a lifelong bacterial geneticist, physiologist, well, mostly lifelong. And actually, she's the daughter of two scientists, and both her parents and Iggy have published in the Journal of Bacteriology. And now she's trying to get her husband to publish it. She started her career at the University of Colorado in Boulder, where she received a degree in biochemistry, and she did her PhD in biochemistry at the University of California, Berkeley, in Bruce Ames's lab, and it's there that she worked on Xtra, E. coli transcriptional regulator that responds to oxidative stress to regulate transcription, and she did really elegant work to determine the mechanism by which Xtra responds to oxidative stress. When she was a second-year graduate student, she just told me, is when she discovered OxyS, which is a small regulatory RNA. And at that time, I don't think anyone really appreciated that bacteria had small regulatory RNAs. And 12 years later, Iggy published her first paper on OxyS, which was also one of the first papers still on small regulatory RNAs. And she has done a lot of foundational work on small regulatory RNAs in bacteria, and she'll talk about that, some of that today, I think. She's gotten oodles of awards, but if I could pick one award I would have wanted, it would have been the Eli Lilly Award, which is given by the American Society for Microbiology to investigator, really promising investigator under 40, and under 40 years of age, and all Eli Lilly Awards, awardees that I know have gone on to have very successful careers. And a couple of years ago, she was awarded the NIH Director's Award. So I think, without further ado, let's welcome Iggy. Thank you for coming.
Thank you. How's this sound? Is that fine? I'm really, really honored to be giving this lecture. Helen Whiteley seems like she was a very interesting and dynamic scientist, and I am often appreciative of sort of the paths paved by women in science before me. I think they've made our lives much easier. But anyway, thank you very much for the honor. What I'd like to do today in my talk is to provide an overview, actually, of several different projects that are going on in the lab related to the functions of small regulatory RNAs and small proteins, which we're arbitrarily defining as proteins of less than 50 amino acids. But before I get started, I sort of want to tell a little story. This is mainly for graduate students. So as Carrie mentioned, I was a graduate student at the University of California at Berkeley, and we did rotations there. When I started graduate school, I thought I wanted to do biophysical chemistry, and rotated with two eminent scientists in Howard Chalkman and Dan Koshland. But then I thought I'd do one more rotation doing bacterial genetics with Bruce Ames. And I found it was really fun. I really enjoyed doing bacterial genetics, but I wondered whether this was it was the right thing to do my graduate work working on E. coli. The project I'd worked on in Dan Koshland's lab was on protein kinase C, which back in 1994 was really hot. And I thought, was I making a mistake to work on E. coli? And my advisor at that time, Jasper Ryan, said, no, there's still things to learn about E. coli. Alternative sigma factors had just been described. So he said, yes, it was fine to do my thesis working on E. coli. And it's sort of ironic because almost 30 years later, I'm still working on E. coli, and I'm still amazed how much we don't know about E. coli. And that is sort of a theme in my talk. And so one of the claims that I'm making is that these genes, the genes encoding small RNAs and small proteins, were genes that were missed. And that's why we haven't studied them, and why not so much is known. And I think it's worth reflecting just a little bit why these genes were missed. And the first is that they were not annotated in completed genome sequences. We are now learning more about what are the features of small regulatory RNAs, but initially it was not, they weren't known, and one could not look for small regulatory RNA genes in completed genome sequences. The same is true for small protein genes. In initial annotation, oftentimes there was just an arbitrary cutoff in terms of the size of proteins that were annotated. In bacteria, often it was 50 amino acids. In eukaryotes, it was often 100 amino acids. That's slowly changing, though a problem now is over-annotation. If you look at E. coli, the annotations of the different E. coli sequences, some E. coli strains have 300 proteins of less than 50 amino acids, and some have 0 proteins of 50 amino acids. So we don't know the exact number in any organism. Some of the other problems why these small genes were missed is that they were missed in genetic screens. They're small, so they're not very good targets. I think another thing that has happened is that people have found mutations in intergenic regions that didn't make sense, and those mutant strains were just thrown out because people didn't know what to do with the mutations or how to interpret them. Small RNAs, and the same is true for small proteins, are missed in biochemical assays. When you run a protein gel, usually run off anything less than 5 kilodaltons. You don't even look for them. But that doesn't mean those proteins don't exist. And initially in microarray analysis, and this is also for proteomic type analysis, things that were not annotated also were missed. This is now changing with new technologies. So we first decided to take systematic approaches to looking for small RNAs. This was, as Carrie mentioned, I stumbled across the oxySRNA as a second-year graduate student, and then was able to publish this work 12 years later. So again, for the graduate students, don't despair. If work you carry out for your thesis doesn't get published immediately, sometimes it gets published 10 years later. But we had characterized the oxySRNA and knew that it acted by base pairing, and this made us wonder how many other small regulatory RNAs there were. And we, together with Susan Gottesman's lab and a number of other labs, have carried out systematic searches for small RNAs. This is now becoming quite popular, actually. The different approaches that were used were to search intergenic regions for stretches of homology between closely related bacteria, searches for promoter and terminator sequences, searches for structure conservation. Now, what is used more frequently are approaches that rely on direct detection of small transcripts, either just by array analysis with probes for the whole genome or deep sequencing. Sometimes this analysis is done after size selection or pulling down RNAs with an RNA binding protein. But in E. coli, the number is not, it's a little squishy, but we usually throw around numbers between 80 and 100 small transcripts have been detected by northern analysis as distinct transcripts. And I think we are making significant progress in learning what these regulatory RNAs are doing. And I just want to provide somewhat of an overview. So many of the RNAs, or the majority of the RNAs, act by base pairing, and this makes sense. And the class that's been characterized most extensively in E. coli are those that act by limited base pairing. So the regulatory RNA is encoded in one location, and its target is encoded in another location, and they share limited complementarity. In many ways, I think they can be seen as similar to microRNAs. And similar to microRNAs, they can repress translation by base pairing at or near the ribosome binding site to block translation. Base pairing can also lead to degradation of the target mRNA, though there are also some examples where the small RNAs have a positive effect on translation. Usually this happens when there's an inhibitory secondary structure that forms in the 5' UTR of the target RNA, and base pairing with half of this 5' UTR opens up the structure to allow ribosome binding. Now in E. coli and a number of other organisms, RNAs that act in this fashion require the RNA chaperone protein called HFQ. This protein is homologous to SM and LSM proteins found in eukaryotic cells. They form this hexameric ring. What is not completely clear, and is actually the focus of a lot of different research in a number of labs, is exactly how HFQ is acting to facilitate base pairing. It's possible, or there's evidence it affects the folding of both the target RNA and the small RNA, and this may promote base pairing. It also binds both the small RNA and the target RNA, so it could bring them into close proximity to allow base pairing. And it's possible that both of these factors are important for some small RNAs and their targets. We also don't fully understand the rules for productive base pairing, what is required to get productive base pairing. But I think what's become clear is shown, as is shown by the examples from E. coli on this slide, is that small RNAs are really integral parts of a number of different stress responses. And this is just a list of some of the different environmental conditions that can induce the expression of a specific small RNA. For example, oxyS is induced by oxidative stress. There's a small RNA that is induced under conditions of anaerobic growth. They're induced by low iron, low magnesium, low temperature. We sometimes wonder if there's a small RNA associated with every specific response to an environmental condition. And usually the situation, as is shown here, is that the small RNA is induced in response to a specific environmental condition. So for example, the Rib RNA is repressed by fur under conditions of high iron, so it's induced when the cells encounter low iron, and then it regulates genes that make sense in terms of the environmental response. So the Rib RNA represses the synthesis of a lot of enzymes that are not essential, that require iron, sort of to allow the iron, the limited iron, to be used by the critical enzymes. The same is true for this RNA that represses genes under anaerobic conditions. It turns off the synthesis of proteins that are not required under the anaerobic conditions. So a lot of RNAs are in this type of network, and it's becoming more and more clear that small RNAs sort of are integral to a variety of different regulatory networks. So there's various groups studying these kind of networks for composed of transcriptional regulators, and they've been given specific names. For example, this is a dense overlapping regulon where multiple regulators regulate a lot of similar genes. there are feed-forward loops. But one question we've been wrestling with or thinking about is why are small RNAs integrated into some of these networks? What is the advantage of having a small RNA? Because there's also similar networks where you just have protein regulators at these different spots where the small RNA regulators indicated in red. Now, we don't have the full answer to this, but Chase Beisel in my group has been studying one feed-forward loop, which involves a small RNA regulator, to try and get at this question, and try to address this question. What is the advantage of having a small RNA regulator in a regulatory network? And the small RNA that Chase has been studying is one called spot42. It was an RNA that was actually discovered very early on as a spot on a gel. It was labeled when cells were labeled with phosphorus. And it was known that expression of spot42 was regulated by CRP, which is a critical regulator of the catabolite repression. And what Chase found when he looked at what are the targets of spot42 is that many of the genes that are repressed by spot42 are the exact same genes that are activated by CRP. So this forms what is called a positive or coherent feed-forward loop, where both the effects of both arms of this loop have the same effect on the targets. So he wanted to get at what are the advantages of having this small RNA regulator in this loop, and he just knocked out spot42 and then looked at what happened to the expression of some of the target genes. And in this case, he took sRNA, one target, and he tagged the protein with SPA so he could examine the levels of sRNA by quantitative Western analysis. And what he found was that when he deleted spot42 and when he went from uninducing to inducing conditions, in a wild-type strain, you have somewhat of a lag that was eliminated or reduced in the spot42 deletion strain. So you're having quicker induction if you don't have the small RNA, which makes sense. The small RNA is keeping the system turned off for longer. Similarly, Chase looked at what happens when you go from an inducing condition to an uninducing condition. And again, when you have the deletion of spot42, you're changing the dynamics. In the wild-type strain, you're seeing a faster shutoff than in the spot42 deletion strain. So the presence of the small RNA is affecting the kinetics of the induction or the repression of the expression of this target. And that's just summarized here. He is seeing the effects under both the conditions of deactivation and the conditions of activation. And this is somewhat different from what has been found for coherent feed-forward loops that involve proteins at position X. And for the case when you have a protein instead of a small RNA, you don't see an effect in the kinetics of deactivation, and then you do see an effect on activation. So the presence of the small RNA, because it's acting at a different point in regulation than the transcription factor would, you're affecting the kinetics of both deactivation and activation. And I think as more and more loops, different networks are characterized that have these base-pairing small RNAs, it will be interesting to see what is the contribution of a small RNA regulator. But for spot42, this is what Chase has concluded. He thinks it reduces leaky expression of non-essential genes in the presence of glucose, and it biases cells to the use of glucose. So the cells stay programmed in state where they want to use glucose, even if you remove the glucose, with the hope possibly that they will again encounter glucose. Chase now wants to do competition experiments where he takes cells and has them grow in glucose and shifts them in and out of glucose and see if the wild type cells outcompete the spot 42 glucose. But in addition to the RNAs that act by through limited base pairing, which is really the class that has been characterized most extensively, there are now an increasing number of RNAs that act by extensive complementarity. These are the true antisense RNAs, where the target is encoded on one strand and the small RNA is encoded on the other strand. And with the advent of deep sequencing, there are reports of hundreds of these RNAs. The problem is that there are very few cases where a physiological role has been demonstrated for these true antisense RNAs. And I just want to describe two that we have been characterizing, two possible functions of these true antisense RNAs, one of which illustrates how we became interested in small proteins. So the first are these small RNAs we call SIBs. This is work carried out by Liz Fozo, who's continuing some of this work in her own lab at the University of Tennessee in Knoxville. But when Liz came to the lab, she agreed to characterize these 5 RNAs that were very homologous, these 5 repeats which expressed an RNA, and she showed that all 5 of these were actually expressed, and she mapped the 5' and 3' ends of these SIB RNAs, which are shown here, and the regions of conservation among these 5 are indicated in red. As I mentioned, Liz carried out northern analysis to show that, in fact, transcripts of 2 different sizes were actually being generated from all 5 of these genes. Now, Liz wanted to figure out what are these RNAs doing? Why are there these 5 RNAs? There didn't seem to be very big differences in their levels of expression. So Liz generated deletions of each of these SIB genes. She deleted each SIB gene individually. She deleted 4 SIB genes. She deleted all 5 SIB genes. And she didn't see a phenotype until she did an experiment in which she introduced a plasmid carrying individual SIB genes into the deletion strands. So what Liz found is that if she had a plasmid where the SIB-C gene was expressed under an arabinose-inducible promoter, she could move this into a SIB-E deletion strain fine and get colonies with and without arabinose, but she couldn't move this into the SIB-C deletion strain. And similarly, she could take a plasmid expressing SIB-E and move it into the SIB-C deletion, but she couldn't move this same plasmid into the SIB-E deletion strain unless she added arabinose. So this result had us baffled for quite a while. What was happening? But it was a little bit reminiscent of a phenotype called plasmid addiction. And this is where plasmids express 2 genes, a toxin and an antitoxin. And the antitoxin is unstable, whereas the toxin is more stable. So when strains lose these plasmids carrying these addiction systems, the toxin and antitoxin, the unstable toxin is degraded, the toxin is around and kills the cell. So then we thought maybe there's a toxin somewhere. This is why we couldn't put the CIB-C plasmid into the CIB-C deletion strain. But where could the toxin be? But we decided to go back and look at this region a little more carefully, this conservation. And we often were struck and joked about the cat-cat sequence. Ha, there's a cat-cat sequence. But if you look on the opposite strand, cat-cat is ATG. And if you look upstream of this cat-cat sequence, there's a CT-rich region, which on the opposite strand would be GA-rich and would correspond to a ribosome binding site. There's also, on the opposite strand, a conserved stop codon. So we realized that on the strand opposite the sRNAs, we are expressing these small proteins, these very small hydrophobic proteins. You can see they're very leucine, isoleucine, valine rich. They're 18 to 19 amino acids. So this is what could explain the phenotype. This small toxic protein was being expressed from the plasmids we were introducing in the cell, and we needed to keep the levels of these small proteins down by expression of the sRNA, either from the plasmid or from the chromosome, to reduce the levels of these small toxic proteins. Liz showed that, in fact, these small proteins are made and are toxic by the experiment shown here. She introduced plasmids in which just this expression of the small protein was controlled by the PBAD promoter. When you add arabinose, the cells are killed. If you introduce a stop codon mutation, then the cells no longer are killed unless you put them in a suppressor strain, which can suppress that mutation. So we are convinced these small proteins are made, and we're also convinced there's really quite a high number of these kind of small protein genes whose expression is controlled or repressed by an antisense RNA in enteric bacteria as well as firmicutes. We're seeing, Liz did a systematic search to look for a number of them. This is just some of them found in E. coli K12. Several of them are repeats. I told you that the 5 CIB genes, we call the protein IBs, are repeated. There are LDR proteins. There's one TISB, SHOB. All of these are very hydrophobic proteins and quite small. What we don't know is what these small proteins are doing in the cell. The fact that they're really quite well conserved, there are 80 or 100 IB genes that are all conserved, to me suggests they have some sort of function. I don't think they are all acting in the same way, because when Liz overexpressed different representatives of these small proteins, she saw different changes in the global gene expression pattern, which is illustrated by these northerns here. Some genes, like the SOX gene, was induced by expression of these 4 different examples. Other genes, like these faded shock protein genes, were only induced by the IBs. But Liz is continuing to characterize these small proteins and their possible functions, as well as their regulation in her own lab. I just wanted to, so one role of the true antisense RNAs, I think, is to control the expression of proteins that have the potential to be toxic. Another role that the small RNAs, antisense RNAs, can do, I think, is exemplified by an RNA we call GAD-Y. This was identified in a search for conserved promoter and terminator sequences, and its expression is induced in stationary phase in an archaeoastronomic manner. So this small RNA is encoded between GAD-X and GAD-W, which encode 2 regulators of E. coli response to acid stress. And the 3' ends of these two genes was suspiciously close, and when we mapped the 3' end of GADX, we saw that in fact this 3' UTR overlaps with the GADY gene. So we observed, and this is Jason Updike, observed that when you overexpress the GADY RNA, but not a control or the antisense to GADY, he saw increased levels of GADX. So when you have elevated levels of GADY, you saw increased levels of GADX. And we have bounced around a number of different models. How could an antisense RNA lead to increased levels of the transcript encoded opposite? But what we think is happening now is that GADY actually directs the cleavage of a longer transcript. So we think that GADX and GADW are transcribed as one long RNA. But this long RNA is pretty unstable. So when GADY is expressed, it base pairs and directs the cleavage of this longer transcript. And the two halves of the transcript are now more stable. So this is how GADY expression can lead to increased levels of the GADX mRNA. And some data to support this is shown here. Jason replaced both GADX and GADW with reporter genes, CAT and GFP, which are more stable, lead to more stable transcripts. In the absence of the small RNA, he could detect a long transcript encoding both of these two genes. In the presence of the small RNA, he would now see the two halves. So we think GADY is directing the specific cleavage of this longer transcript, and think similar things could be happening at other genes that have an antisense RNA, and this could have a number of different regulatory consequences. So one can imagine, as in the case of GADX and GADW, you have one long transcript that is not very stable and the two halves are more stable, or you could have it such that the long transcript is stable and the two halves are less stable, and various permutations of this. So these are, I think, two possible roles for these cis-encoded antisense RNAs. They control the expression of potentially toxic proteins, and in the case of GADY, direct the cleavage of a longer transcript. Now, although base pairing, regulatory RNAs that act by base pairing have really been the focus of most of the work on regulatory RNAs in both bacteria and in eukaryotes, I think it's important to consider that RNAs can act in other ways. RNA is a pretty flexible molecule, and in bacteria there are two good examples of RNAs that act by mimicking the structure of other nucleic acids to modulate the activities of proteins. And I think there will be additional examples of RNAs that act in that way. But just to mention two examples, Karen Wasserman, first in my lab and now in our own lab, has been characterizing one RNA called the 6S RNA, which mimics the structure of an open promoter. And 6S RNA is actually a good example of why RNAs were missed. So 6S was one of the first nucleic acids to be sequenced. Its sequence was published in 1971. And it was known to bind to proteins, but we didn't know what proteins it bound to until Karen carried out a glycerol gradient and showed that it bound to subunits of RNA polymerase. Now, when Karen first showed me this result, I didn't quite believe her. This was binding to RNA polymerase. Hundreds of labs study E. coli RNA polymerase. Why had no one ever seen this RNA? And the reason is the first step of the standard RNA polymerase purification is designed to remove nucleic acids. So people do a high salt wash or polyamine P precipitation, and it was designed to remove the nucleic acids, and everybody follows the same purification protocol, which is why success was missed. But Karen has gone on to show, and our other labs are also characterizing success, that it inhibits the sigma 70 form, or the housekeeping form of RNA polymerase under stationary phase conditions. Another RNA that acts by mimicking another nucleic acid is CSRB and other members of the CSRB family of regulatory RNAs. Now, all of these RNAs carry multiple binding sites for a protein called CSRA, an RNA binding protein called CSRA. And CSRA binds to the 5' UTRs of various target mRNAs to repress translation or activate translation or affect message stability. And CSRB and its homologs acts by titrating the CSRA protein away from its target mRNAs. But as I mentioned, I'm suspicious that there are going to be other RNAs that act by binding proteins and mimicking other nucleic acids. And my favorite candidate right now is RIFE-D. And I'm mentioning this because I'm trying to advertise this RNA. I'm dying for a postdoc to come work on this RNA. But RIFE-D is predicted to fold into a very base paired type of structure. It's encoded in a region between two genes in sort of an unusual way. The promoter for RIFE-D is within the upstream gene, and RIFE-D actually contains part of the CLIP-B mRNA. But Microbiology, when he was in the lab, showed that RIFE-D binds to proteins. If you fractionate cells on the glycerol gradient, then you can see in the absence of phenol treatment, the RIFE-D RNA runs differently than if you extract the protein, so it appears to be binding to proteins. And Lori Waters went on to carry out sucrose gradients, which suggests that the RIFE-D RNA is associating with polysomes or the 70S ribosome fraction. So I'm intrigued by the possibility that there could be regulatory RNAs targeting the ribosome, which is itself is comprised so much of RNA. So it seems to me that could be another target of regulatory RNAs. But we clearly have more experiments to do to test this possibility. So I also want to point out, with all of the reports of small RNAs, the question of whether all transcripts have function. If one can detect a band on a northern blot, does that mean it will have a function? And again, I don't know the answer to this question, but when we carried out sequencing of cDNAs derived from size-selected RNAs, we obtained a lot of sequences that corresponded to the 3' ends or 3' UTRs of messages. And some examples are shown here. So, for example, for the FIN-A message, here is the transcript, and here we see a fragment from the 3' end. And we see a lot of this band. It's a really defined band. Similarly, we see very clear bands coming from the 3' ends of these different genes. Will these small transcripts, which can be considered small RNAs, have functions, or are they just very stable products that arise from the degradation of these mRNAs? And I think that's something to keep in mind with the reports of these hundreds of regulatory RNAs. Do they all have function? I think the onus will be on showing that, in fact, they do have function. And now I want to turn to the last part of my talk, and that is the question, are all small RNAs non-coding? And clearly, the fact that I call these RNAs small RNAs and not coding RNAs indicates that I think that they are not all non-coding. We have now come across a couple of different examples where we thought it was a regulatory RNA and we found a small protein. And one example is an RNA that was initially called RIBE-A. So it was encoded we knew it was encoded in this intergene region between YLIL and MNTR. So Lori Waters mapped the 5' and 3' ends of this gene, and when she did an alignment and looked at the sequence more carefully, she realized that this small RNA actually encoded a protein. Here's the open reading frame, and you can see that the region that is conserved really corresponds to the conservation of this small protein. So instead of being a small regulatory RNA, we think this small transcript encodes a small protein, which we are calling MNTS. Now we've been able to learn about this MNTS protein because of a clue. And we noted that MNTS was encoded opposite a regulator of the manganese response. So it was encoded opposite the MNTR regulator, and if you looked at the promoter sequence, there were MNTR consensus binding sites. So Lori tagged the open reading frame, the 42 amino acid open reading frame, and showed that MNTS clearly is regulated by manganese in an MNTR-dependent manner. So in minimal media, you see high levels of the protein. When you add manganese, you get strong repression. In rich media, you have lower levels of the small protein, which increase when you add ETTA, which presumably chelates some of the manganese that's present in LP. And so we now propose that MNTS is a chaperone for manganese, that it's a protein that helps deliver manganese to the appropriate proteins in the cell. And some of the data which makes us believe this is shown on the next few slides. One experiment Lori did was to take the nickel affinity columns that are used to purify his-tagged proteins and load these columns, this resin, with manganese instead of nickel. And she found that MNTS was retained on these beads, whereas a controlled protein was not. Lori also found that if you delete MNTS, you affect the ability of MNTR, which binds this repressor, which binds manganese to repress a different target. This is the MNTH target. In wild-type cells at 1 micromolar manganese, you see strong repression, which you don't see. You see less complete repression in an MNTS mutant. And the most dramatic phenotype came when Lori overproduced the MNTS protein. And she saw that cells that overexpress MNTS have a specific hypersensitivity to manganese and not to other metals such as zinc or magnesium or iron or copper. So this protein seems to specifically affect manganese metabolism in the cell, and we are now collaborating with Jim Imlay and Joan Stubby's lab to try and show that it is in fact required for the manganese loading of specific enzymes. But it made us think about how many other proteins of less than 50 amino acids are missed have been overlooked. And we arbitrarily took a cut off of 50 amino acids. We decided it was worth trying to pursue finding more small proteins, because some of the small proteins described in the literature have interesting function. There are examples of small proteins that are self-density signals. In bacillus, there's a small protein SDA, which inhibits the activity of a kinase. There's also examples in eukaryotic cells of small proteins that have important functions. So we thought it was worth trying to more systematically look for small proteins. And we used the same approach, so we went back to the intergenic regions that we scanned for the small regulatory RNAs. We looked for conservation, but in this case, the conservation that we were looking for was different than the conservation we were looking for the small RNAs. We were looking for conservation of AUGs and stop codons, as well as synonymous changes and conservative substitutions, as is shown here. So we searched for homology, for ribosome binding sites, for homology in combination with ribosome binding sites. And then what we did, which I think was important, was to tag all these small open reading frames at the C-terminus with the SPA tag. This is a double tag, a flag, and module and binding protein tag, to see that these small proteins were synthesized. And we showed that 20 previously annotated, as well as 18 newly discovered small proteins were in fact synthesized. And some data to show this is shown here. For example, this one, YOHP, the expression of this is induced in stationary phase. You can see different ones show different kinds of regulation. So these small proteins are being made in E. coli. What surprised me is that over half had features suggesting that they have a single transmembrane domain. So they have a hydrophobic sequence predicted to form a single transmembrane domain. And when Matt Hem, who carried out this work, fractionated cells, he showed that, in fact, these small proteins, as predicted, fractionated with a membrane pellet. That doesn't mean that none of them are cytosolic. There are some that fractionate with the supername, but the majority seem to be in the membrane. And this is not something I expected. Now the big question is, what are all these small proteins doing? I think there's a lot of interesting questions. How do they get out of the ribosome? How do they get inserted into the membrane? What is their orientation in the membrane? But a critical question is, what are the small proteins doing? And we're trying to use every possible approach to get at their functions, and I just want to briefly mention two examples. One protein is YBHT. It's on the large side of these small proteins. It's always found adjacent to genes involved in forming molybdenum. But Eric Hobbs was able to get clues into the function of YBHT using both a barcoded deletion set and looking for phenotypes and pulling down the SPA tagged version of this protein. So Eric knocked out all small RNA genes and all small protein genes by inserting unique sequence tags, so you could mix all these deletion strains and look for strains that were sensitive or resistant to a specific stress condition. Here he looked at sensitivity to cell envelope stress, and among this collection of deletions, he found some that were very sensitive to cell envelope stress, others that were resistant. One that was sensitive to the cell envelope stress were strains carrying a deletion of this YBHT gene. But when Eric pulled down the YBHT protein, you can see it here, after elution from a flag affinity column and a calmodulin affinity column. He saw YBHT, but he also saw two co-purifying bands, which correspond to ACRA and particularly ACRB. So there was a strong binding between YBHT and ACRB, and Eric has in fact shown if he pulls down ACRB, he can co-purify YBHT. What are ACRB and ACRA? They are components of a major efflux pump in bacteria. So the ACRA-B-Tol-C pump, proteins pump out drugs from the inner membrane and periplasm out of the cell. They're critical for the resistance to a number of different antibiotics. And Eric found that if you delete YBHT, you also are sensitive to a number of antibiotics, though not to the extent that ACRA and ACRB mutants are sensitive. So we think that somehow YBHT modifies this exporter. Exactly what it's doing, whether it's affecting the assembly of the exporter, or my favorite model is that maybe it changes the specificity of the compounds that are exported by ACRB, ACRA-B. And I'm really intrigued by the possibility that molybdenum, if you squint at it, doesn't look that dissimilar from some of the things like tetracycline that are exported by ACRA-B and to which YBHT mutants show sensitivity. So one more example of a small protein that interacts with an exporter or a transporter is provided by YNEM. This is a smaller protein. It's 31 amino acids. It's encoded next to a regulatory RNA gene. And in this case, we obtained some clues to YNEM, to the YNEM role by looking at the effect of overexpressing YNEM. And what Eric found was that when he overexpressed YNEM and looked at whole genome expression, a number of targets of the FORB phosphate regulon were induced, as is shown here. And Eric showed that a specific target, a reporter for the FORB two-component system is in fact induced by overexpression of YNEM. So here he monitored affin C, lac C fusion, which normally under these growth conditions has very low activity. And if you induce YNEM, you get induction of this fin C, lac C fusion. Since so YNEM affects this FORB two-component system, and we're intrigued by the fact that expression of YNEM itself is regulated by another two-component system, the FOQ, FOP two-component system, whose activity is regulated by low magnesium or acidic conditions. So here we have a small protein whose expression is induced via one two-component system, which somehow modulates the activity of another two-component system. And there's an example of another small protein. This is work carried out in Mark Goulian's lab, which has shown that the MGRB small protein affects the FOQP two-component system. So there's an example, and I think this will be a theme, that small proteins might be modulating the kinases of the two-component systems. Because there's evidence that MGRB interacts with FOQ, we really thought that YNEM had to interact with FOR. So we tried very hard to see a physical interaction between YNEM and FOR, and we have not been able to show this. We don't think these two proteins interact. What we think instead now is that YNEM might affect phosphate transporter. Phosphate transport through these phosphate transporters, in particular PIT-A. So PIT-A and PIT-B are two proteins that can, at low affinity, bring in phosphate. And when you make a deletion of PIT-A, you no longer see induction of the thin C fusion in this mutant. So you see induction in the wild-type strain in the PIT-B mutant strain, but not in PIT-A. We also, in an assay where we cross-link proteins with YNEM, we see a complex in wild-type cells. This is a cross-linked product where we think we have YNEM cross-linked to another protein. And this is no longer seen in a PIT-A mutant. So we think that maybe the YNEM small protein is interacting with the PIT-A transporter, providing yet another example of a small protein interacting with a transporter, possibly somehow affecting its activity. But let me stop there. Since the small protein project is really at its beginning, it's, I think, where we were with small RNAs about 10 years ago, it's fun to speculate. So at this point, we can speculate about anything these small proteins can do. I like the idea that they modulate transporter specificity or assembly. They could modulate the activity of regulatory proteins, function as part of a multi-protein complex, function as chaperones. We think MNDS is a metal chaperone. They could modulate or sense the character of the membrane, form a pore, act as a signal. We can keep going on without data, but I think it's fun to think about what the small proteins are doing and how many more are out there and have been missed. But let me end with just a brief summary. So for small regulatory RNAs, one class acts by limited base pairing, and these form integral parts of a number of different regulatory networks. There are a few small RNAs that have been characterized that are true antisense RNAs that block the expression of potentially toxic proteins and direct mRNA target processing. Some RNAs act as regulators by mimicking other nucleic acids. I didn't go into this in detail, but the elucidation of a number of small RNA function has solved some regulatory mysteries. But it's exciting. There's still, the roles of many small RNAs still remain to be elucidated. And this is even more so for the small proteins. We can definitely detect small proteins in E. coli. They're made. We have some clues about regulation, still some deletion phenotypes and overexpression phenotypes, and co-purifying proteins. So I think we will be able to learn more and more about the function of the small proteins. There is a subset which is toxic at high levels. But this is the most exciting statement. There's a lot more to be learned about the small proteins. And let me end with listing all of the collaborators. I've tried to mention the members of my lab who've carried out this work throughout the talk. The people in blue are working on the small RNAs in the lab. The people in purple are working on the small proteins. You can see Lori is working on both. These are former members of my lab who worked on either the small RNAs and small proteins. A number of these are now continuing studies in their own labs. And we've had very terrific collaborations with scientists at NIH and elsewhere. Thanks again for your attention and the honor, and I'd be happy to answer any questions.